Hello, students. Today, we're going to learn about some really amazing ways that living things, especially plants, can reproduce all by themselves. It's a special ability, and we are going to explore it together.
So let's start with a question to get you thinking. We all know that plants grow from seeds, right? But what if I told you that a part of a plant, maybe just a piece of its root or even a single leaf, could grow into a whole new plant? Is that possible? Let's find out.
This incredible ability actually has a name. It's called vegetative propagation. So this is our first topic, and we're going to see exactly how it works. Okay, so what is this vegetative propagation? Well, the definition is right here. It's a type of asexual reproduction. And the most important part to remember is that new plants are produced from the vegetative parts of the parent plant. That means its roots, its stems, or its leaves. Simple, isn't it?
Now take a look at this. This is a fantastic example. This is the leaf of a plant called bryophyllum. Do you see those little plantlets growing right on the edge of the leaf? These are tiny buds. If they fall on moist soil, each one can grow into a completely new bryophyllum plant. Amazing!
And you see this method being used for many common plants. For instance, a potato. Those little eyes you see on a potato, those are actually buds. If you plant a piece of potato with an eye, a new plant will grow. Similarly, farmers grow sugarcane and roses just by planting cuttings of the stem. Even plants like banana and jasmine, which can no longer produce viable seeds, are grown this way. So, you might ask, why is this method so useful for farmers? Well, there are two main reasons. First, it's much faster. Plants grown this way will produce flowers and fruits much earlier than plants grown from a seed. And second, all the new plants are exactly like the parent plant. This means if you have a parent plant with very sweet fruits, all the new plants will also have very sweet fruits. The good quality is maintained. Now, what happens when we take this same idea and we use it in a modern, high-tech laboratory? Well, we get a method called tissue culture. So how does this work? It's quite a neat process. First, scientists take a tiny piece of tissue from the growing tip of a plant. They place these cells in a special gel, an artificial medium, where the cells start to divide and form a lump called a callus. Then, this callus is transferred to another medium that contains hormones, which tell the cells to grow into tiny plantlets with roots and leaves. Finally, these little plantlets can be planted in soil to grow into mature plants. And the biggest benefit of tissue culture is that you can grow thousands of plants from just one parent, and you can do it in very clean, disease-free conditions. This technique is often used to grow ornamental plants, the kind you see for decoration. All right, but plants aren't the only ones that reproduce asexually. Let's now look at another method called spore formation. You have probably seen this on old bread. It's very common in fungi. Yes, this is the bread mold, also known as rhizopus. I'm sure if you've left a slice of bread in a damp place, you have seen this fuzzy, thread-like growth. If you look closely, you can see these fine threads and some tiny blob-like structures on top. Let's break down what we are seeing here. The thread-like parts are called hyphae. These are not involved in reproduction, but those tiny blobs on top, those are the important part. They are called sporangia, and they are like little packets filled with tiny cells called spores. These spores are what grow into new bread mold. Now, the most important thing to know about a sporia is that it has a very thick, protective wall. This wall acts like a shield. It protects the spore when conditions are bad, like if it's too dry or too hot. And when the conditions become good again, when it finds a moist surface, it can start to grow. So let's quickly compare the two methods to make things very clear. Vegetative propagation uses parts of the plant's body, its root, stem, or leaf. Spore formation, on the other hand, uses special cells called spores. Vegetative propagation is common in higher plants, while spore formation is common in organisms like fungi. And a very important difference, spores are designed to survive bad conditions, whereas a plant cutting needs good conditions to grow. Now here is a question that confuses many students, so let's clear it up right now. Are spores the same thing as seeds? The answer is a very clear no. They are not the same. Please remember this. Spores are single cells and they are formed from one parent through asexual reproduction. Seeds, on the other hand, are much more complex. They contain multiple cells and are formed from two parents through sexual reproduction. All right, we have covered a lot today. So now let's just calmly go over the most important points that you should remember for your exam.
So let's summarize. First, we have vegetative propagation, where new plants grow from roots, stems, or leaves. It's a fast method and produces plants that are just like the parent. Remember the examples: potato, rose, and bryophyllum. Then we have tissue culture, the modern lab method for growing many disease-free plants. And finally, we have spore formation, used by fungi, which involves tiny spores that can survive harsh conditions because of their thick walls. And to end today's class, I want to leave you with a question to think about. Between vegetative propagation and spore formation, which method do you think gives a species a better chance for long-term survival? And why do you think so? Ponder on this. We will discuss it next time.